There are 7 types of pleiotropy. The pleiotropy can be basically classified into 7 types which is given in the table. These have different levels of importance and significance. All these types of pleiotropy carry a long term retribution in terms of evolutionary constraints. A gene controls more than one biological function is more limited in its envelope of possible variations than is a monofunctional gene. Moreover, alleles that is selectively advantageous for function in one tissue may be disadvantageous to function in another tissue. Here you can see a table wherein classification of different types of pleiotropy is given. The first column gives the type of pleiotropy, the second column gives the situation, and in the third column the example is provided. Now let us look into each type one by one. The first one is artifactual pleiotropy. In this case, a single mutation can affect more than one process because 2 genes are located next to each other in the genome. An example is the claret non-disjunction alleles in Drosophila, which affect both meiosis and eye color. The claret locus of Drosophila melanogaster comprises 2 separately mutable domains, one responsible for eye color and one responsible for proper disjunction of chromosomes in meiosis and early cleavage divisions. There are 3 types of alleles. 1. Alleles of the claret type that affect eye color only. 2. Alleles of the claret non-disjunctional type that affect eye color and chromosome behavior. And 3. A meiotic mutation non-claret disjunctional that affects chromosome behavior only. The behavior of claret alleles was cleared when it was cloned. There is an adjacent gene which encodes a kinesin molecule required for normal chromosome disjunction at meiosis, and the pleiotropic alleles are small deletions that affect both transcription units. Therefore, the multiple effects of such mutations are artifacts of the process of mutagenesis, hence the name. Especially the organisms with compact gene dense genomes are susceptible to artifactual pleiotropies. Now let us look into the second one, that is secondary pleiotropy. This is equivalent to relational pleiotropy. It describes the situations where a simple biochemical abnormality has multiple phenotypic consequences, sometimes with connection to the initiating mutation. Long-lived complex organisms, especially those with advanced abilities to compensate for physiological dysfunction, exhibit secondary pleiotropies. For example, one of the first human mutants is provided by the disease phenylketonuria to be understood at a biochemical level. In this case, a defect in phenylalanine hydroxylase, the liver enzyme, that leads to excess plasma levels of phenylalanine. In the brain, it affects myelination of axons and finally to mental retardation. Similar or more complex chains of developmental and physiological consequence can be seen in many genetic disorders in humans, such as the complex syndromes arising from thalassemias. Rudimentary mutations affecting pyrimidine biosynthesis, a familiar example is provided in Drosophila. Some of these mutations lead to defective development of the wing, but not of the rest of the fly, because pyrimidine levels become rate limiting for cell division in the rapidly proliferating tissue of wing disc, but not elsewhere. The third one is adoptive pleiotropy. It is otherwise known as exaptational pleiotropy. In this, a pre-existing protein has been clearly exapted or co-opted in evolutionary terminology to implement an additional function that is not related to its original biochemical role. An example of this type of pleiotropy is crystalline proteins, which is the most abundant proteins in lens tissue and play a refractive and structural role. It is a protein that in humans is encoded by the CRYGD gene. 4 gamma crystalline genes, gamma A through gamma D, and 3 pseudo genes, gamma E, gamma F and gamma G, are tandemly organized in a genomic segment as a gene cluster. In many processes, this protein acts as familiar metabolic enzymes such as anolase and LDH. Now, let us look into parsimonious pleiotropy. Parsimonious pleiotropy encompasses cases where the same enzyme catalyzes the same chemical reaction in multiple different pathways. Example, human guanylate binding protein, in short we call GBP1. GBP1 is a multi-step regulator of CAMP-PKA signaling, inhibits PKA downstream of GPA2P and CYR1P, thereby increasing CAMP dependency, promotes ubiquitin dependent proteolysis of IRA2P regulated by G alpha protein GPA2P. GPB1 has a paralog GPB2 that arose from the whole genome duplication. Complex metabolic consequences occur because of the loss of this enzyme. For example, at several steps in the biosynthesis of isoleucine and of valine, the same proteins are used. So, in bacteria, knocking out any of these enzymes leads to simultaneous prototrophy for both amino acids. In such cases, there has probably been no pressure to evolve independent regulation of the two pathways, either at the enzyme level or at the gene level. So, the organism utilizes the same protein in each pathway. Now, let us look into fifth one, opportunistic pleiotrophy. It is related to the two preceding types. A regulatory protein is recruited to perform an additional function in a different tissue. Example is the cis-B ASC genes acting as numerator elements on the X chromosome of Drosophila. In Drosophila, sex is determined by the ratio of X chromosome dosage to autosomal chromosomal dosage, and a few sites on the X chromosome act early in development as major numerators in setting this ratio. The sisterless A, in short called as cis-A gene, functions as one of 4 X:A numerator elements that set the alternative male or female regulatory states of the switch gene sex lethal. In somatic cells, cis-A functions specifically in sex determination, but its expression pattern also hints at a role in the yolk cell, a syncytial structure believed to provide energy and nutrients to the developing embryo. By genetic criteria, cis-A and cis-B are the most potent zygotic determinants of X chromosome dose. Now, let us look into the sixth one, combinatorial pleiotropy. Combinatorial pleiotropy applies in many of cases where a single protein interacts with a variety of different components in different cell types, and it altered biochemical activity and specificity in each different situation. This protein will have potentially diverse and multiple effects on a variety of tissues when affected with mutation. The same combinatorial strategy is observed even in unicellular eukaryotes. For example, in Cyanorhabditis elegans gene UNC86 is required in several distinct neuroblast lineages for daughter cells to become different from their mothers, and is also required for the specification of particular neural identities. Another example, the proteins that control mating type of yeast include the DNA binding protein encoded by MAT alpha 2, which has different functions in diploids and alpha cells. In diploid cells, it acts in combination with MAT alpha 1 to repress an additional set of haploid specific genes, but in haploid alpha cells, it acts to repress alpha specific genes. Now, let us look into the case of seventh one, unifying pleiotropy. Unifying pleiotropy is the final type because in ultimate biological output, it describes cases where the multiple functions of genes or locus are all related, but the immediate chemical functions are diverse. The different enzyme activities, structural components or binding domains are included in the same polypeptide chain or encoded by adjacent cistrons under common regulatory control. Otherwise, mutation of the locus can have complex physiological consequences. For example, in Cyanorhabditis elegans, the gene CHA1 is the structural gene for choline acetyl transferase, the enzyme which synthesizes acetylcholine. CHA1 is a complex gene which includes in UNC17 locus. It has been hypothesized that a single protein is encoded which consists of several discrete structural domains.